So welcome everybody on behalf of Center for Research on Complex Generics. I'm very happy to welcome you to our very first in-person workshop. We hope many more will be happening this year and over the next years. So we're happy to see everybody in person and alive after pandemic and doing so well. It's a very important workshop. It's around considerations for alternative comparable clinical endpoint and pharmacodynamic equivalence studies for generic orally inhalable drug products. It's a very big area, and we will highlight and really hopefully learn a lot in the next 2 days around it. You probably know by now, Center for Research in Complex Generics started in 2020 based on the grant, U01 grant we received between University of Michigan and University of Maryland from FDA. Our mission is to increase access to safe and effective generic drug medication through 3 main goals of the center around communication, education, and collaborative research. It's really had been a very good partnership between 2 universities, FDA, and many generic companies and other stakeholders who put their input into the main topics we should be focusing on, our research, education, and what are the real challenges around development of those complex drug products. So as I mentioned, we have kind of 3 main pillars of our effort. It's one of them is to develop infrastructure and to have communications. And through it, we really interview a number of players in the generic industry, small and large, and some CROs to other partners and stakeholders to understand where the issues truly lie. And through those interviews, orally inhalable products come up over and over again, and the need for alternative studies. The second mission is around education and training, and this is why we all are here. We're gonna listen to research and talks around it, and hopefully we could pave out the way for the development of new approaches over the next couple of years. And then we do some of the small collaborative research projects, some small pilot projects, and over the years, we hopefully will also fund larger projects through the center if the funding increases. Through our interview process, we approach quite a number of companies. Maybe not all of them are here, but some of them are very large generic companies like Amneal, Apotek, Dr. Reddy, Fresenius, Teva, Sun, and there are some smaller companies, but there are also some stakeholders like AAM and Pharma, Biopharma, IPEC-RS. So through those continuous interviews, we constantly learn about new problems the industry is developing and what are the issues. And if you guys here or online would like to schedule a meeting with us, please do contact us and we will be happy to start the dialogue. So why we are so excited about this workshop, today's workshop, is through this interview, we understand that orally inhalable products, those are huge products. They're all billion-dollar products. So many companies around the world are very interested in developing alternative generic versions for many of those products. And with the COVID-19 pandemic, you could see that we will have more and more need for orally inhalable products in the future. So there are new being developed in the innovator side. They're very unique with its own unique challenges. Almost the entire pharmaceutical sciences field is around one inhalable product. They're often insoluble drugs. They have require complex characterization of particle size, dissolution. There are not many standard analytical methods how to define all of those parameters. There is quite a bit batch-to-batch variability in innovator products. there are drug-device combinations. So the devices also impact how they're used. There is substantial patient variability in how devices are used and populational differences in pharmacokinetics and efficacy. So because of all those unique challenges, really currently very large endpoint studies are required for approval of generic versions, and often the cost of endpoint studies is higher than the cost of the approval of innovators, as many of you did mention. And it's all because of this large intra and inter-patient variability. And so many companies really would like to have discussed the design and clinical trials before they invest in them. So all of it really highlights the need for alternative approaches. And it's, again, a very big problem, and hopefully over the next several years, we could come up with some good approaches when we could use a combination of in vitro characterization and in silico modeling, and hopefully reduce the burden of the clinical trial cost and have many more of those medications accessible. Okay. Okay, it's stuck again. Okay, no. So as a center, we do plan several workshops, and hopefully there are many, many FDA attendants here. So everybody who are on virtual, please do come in person. You will have a chance to sit in a room and to have this discussion. On May 10th, we have a really cool workshop around drug-device combinations, and June 15th, nitrosamines and other big topics. And then there is PBPK modeling workshops and other drug-device combination by equivalence, and also another one highly important that comes up a lot is characterization of complex excipients and formulations. So please keep them in mind and hopefully attend. As far as the research, those are the listings of the small research projects we have at both universities, some of them around the specific products like irinotecan liposomes, polymer-based PLGA, some of the meter-dose inhaler products, and then some evaluation of different dissolution media for oral absorption. As a team here, we all are. Jim Polly at University of Maryland and myself, we co-lead the center. Visha Krishnan, who you all know, she is the manager. And then Jen Dick is administrative assistant, and Dana Hamill making sure that everything runs very well. So we all appreciate all of their help with this workshop. And then if you want to get a hold of us, here is all the way you could find us through the website. We have a YouTube channel where we post all of those recordings, and we have a presence on social media on LinkedIn and Twitter. So please do follow us, connect with us, and if you need to contact us for an interview, let us know. With that, I really would like to welcome the next speaker, who is Rob Lionberger. Rob is the Director of Research Standards in the Office of Generic Drug Products. And Rob had been with FDA 20 years, over 20 years, and Rob had been instrumental in starting our center. He, the one who proposed the need for a center like that, and we are very grateful for it. And his degrees are in chemical engineering from Stanford and Princeton, and then he's been working in the area of modeling and simulations and leading the organization. Welcome, Rob.
All right, thank you, Anna. It's really a great pleasure to welcome you to the. All right, well, they're working on my my slides. But this is our first CRGC workshop that's in person. So I'm really excited to be here with all of you for that event. And I think this is part of our vision for the Center for Complex Generics. It's to really have in-person events that have the key players in the generic drug development, in the room together with the FDA staff, talking honestly and clearly about the scientific challenges that we face in generic drug development. And hopefully this interaction can facilitate, as Anna said, access to generic versions of complex products. All right, so why are we here? So let's start with just the basic fact of this, is that there are about 33 MDI, DPI products that are approved and actively marketed. If you look at our FDA's Orange Book, only 4 of them have available generics. All right, so this is where the opportunity is. This is where the value creation lies. This is the reason for this workshop. And so if you think about this number, some people think that the glass is half full. Some people think the glass is half empty. So, the first success is this number, the fraction of products that have generic competition, it's not 0. Like, that's a success, right? If you go back to 2018, this number was 0. There were no generics available. So we have provided a pathway that's viable. You can get products approved using the approach. Have a few key landmarks here that we've seen the first approval. We've seen some places where there's beginning to see multiple approvals and a sort of viable competitive framework. So I think this is, and it took a long effort to get there. Those of you who are involved in these applications, submissions, and approval know how costly and time-consuming these development programs were. And if you think about that, really the goal of this workshop is we can do better than 4 of these. And the one thing that's going to enable that is more efficient approaches to bioequivalence. Like, that's the key burden. If you're all the different things you have to do to match up these products, there's a lot of opportunity, there's a lot of markets, but getting access to those really is going to require more efficient bioequivalence methods to be implemented there. And that's really the focus of why we're here today. All right, and I want to just be very honest and very clear about what the current state is. And this is, my personal view of the current state of our scientific aspect, is there are clearly better approaches to bioequivalence than FDA's weight of evidence approach. You'll hear this morning about the clinical endpoint data that we've seen. I think everyone who works in this field knows that the dose-response relationship for those studies is extremely flat. You could have big differences and still get the same clinical response. That's a fact of this field. And I think that's why it's very clear to say that an approach that combines in an appropriate way in vitro comparisons and analysis of PK studies can offer just as good or better assurance of local delivery of drugs to the lung than the current approach. And I think that's, that's my scientific opinion, but I think that's based and backed up by all the work that's, it's happened. If you read the literature, if you think about these very carefully, that's true fact. However, where we are in the regulatory science of this is the actual implementation details aren't exactly clear. Well, exactly what's the best way to analyze a PK study to learn about local delivery? Which of the various in vitro tests are the most efficient way to demonstrate bioequivalence? We've advanced our particle characterization a lot. What role does that play? We have new mouth-throat models. They're more predictive. How do we use those? We have different methods for dissolution of particles, right? None of these things are in FDA's current weight of evidence approach for bioequivalence, but which one of these should be in a more efficient bioequivalence approach? Which are too sensitive? What's the relationship? Even when you pick the right one, if you say, look, I'm looking at sort of particle size and surface chemistry, morphology characterization, the better I get at my analytical methods, I'm going to see differences between 2 products that come from different manufacturers. When are those differences significant or not? So there's a lot of implementation challenges. There's a lot of value that can be created by, you'll hear a lot about quantitative methods at the workshop today, both from the PBPK type models to help interpret the PK studies, as well as the CFD models to help understand the different deposition products. And I think those can be incredibly important to building out these implementation details. What's the relationship between I measure a difference and whether that's going to have a clinical impact? The modeling approaches, as we make them better and better, can help understand that sensitivity connection and help put together this full picture. And again, just to be very clear, like we in our guidances, in some of our guidances on inhalation products, we've pointed toward alternative approaches. We've encouraged people to meet with them, but we haven't at this time can't go through and give you, here are the 5 different studies you do for the alternative approach. And if you do these 5 studies, you're going to be on a very clear path to approval. That's really our standard for updating our guidances. And so where we are now is we think the scientific promise of the alternative approaches is there. The details we can't give you at the level of clarity that we think the guidance would have to have. And so what do we do in that situation? All right, if we can go back one. All right. So we're going to give you two things that you can do here. One is we have since GDUFA 2, now in GDUFA 3, we have the opportunity for generic applicants to meet with us in what we call product development meetings. So these meetings are intended to address this exact situation where you want to propose an alternative to what FDA says in the guidance that's more efficient. And we really strongly encourage, if you're in a development program, you want to pursue and get feedback on this, use these meetings extensively. Use them at the beginning. We're happy to meet with you at the beginning. Oh, I'm working on a modeling approach. Let me get some feedback on the modeling approach. I want to make sure that that's working well. And then maybe I generate some initial PK or characterization data. I want to walk through and talk through that data and understand, is that method going to be convincing now that I have this model that helps me set some limits? Meet with us on that topic. And then as you get closer to submission, have a pre-submission meeting where you go through the full alternative approach and outline it for our reviewers. Really leverage this. I think this is incredibly important to getting, product-specific clarity on these new approaches. And so I think that's the important, if I ask you to do one thing, it's really engage through this meeting process on these development programs to get the feedback earlier, and it won't be a situation where you end up with a 100-page deficiency in your application. Right? Engage early on the meeting process. I think that's important for moving these novel approaches forward. And and I think it's our intention. If there's any viable approach in this space by our commitment letter rules, these all fit into the alternatives to existing guidances. So FDA will grant your meetings for this. So there's no sort of, oh, will FDA grant, not talk. We will. That's part of our commitments. And so we really want to see and engage in that. And think about we don't want these meetings to delay your development program. So think strategically about how you submit the meeting packages, what topics you want to talk about at different points in the development so that it can progress. So it's not we don't want to have a situation where you're waiting for the meeting and then you do the next step. So you have to, plan and, understand the timelines and understand the engagement. But we're happy to engage with you here in some of the Q&A sessions on what the best way to do that is, what the way to break up packages into different topics at different points in development, as well to make the meeting packages, the meeting process work for you. So that's the first step. The second step is things like this. I mean, the CRG workshops, the CRG was really focused on providing a forum where we can have these type of interactions, where there's a promising scientific area, but the implementation details into your development programs and our regulatory science programs are still uncertain. And so we want to bring together people who are actively working in this. We hope through the panel discussions, the presentations from industry stakeholders, that we'll really mutually understand like what's difficult to do, what's uncertain. If there's 2 different ways to do something, get a better sense of what's the most appropriate way to do this. Again, there's lots of, some of the things that we have funded and others have developed, lots of new approaches to characterizing these. Which mouth-throat model is better? How does that fit into our development program? If FDA's current guidance says use a cascade impactor, but I want to use a more realistic mouth-throat model as part of an in vitro alternative approach. What are the details there? What do I see in those teams? What are the details of implementing that? Are these tools available? What if I'm using approach A versus approach B? Those type of technical details, we really want to have detailed discussions here to help hash that out and help develop, in the public way, the thing. So you really have two options. One is there's the private product-specific approaches through the meeting process, for the proprietary aspects of it. But there's also the public aspects of the community developing the tools and the best practices around those tools that, can also be much more efficient, help us move toward more specific guidance for these. So I really look forward to today's events and engaging with the people that are here. Also encourage people who are participating remotely to send in your questions so we can have them reflected in the discussions, provide you opportunities to ask questions to the speakers as well. And hopefully this workshop will help advance and move forward toward more generic competition for this important product category. So really look forward to today's events, and thank you all very much for being here.
All right, good morning, everyone. I want to welcome all of you to day one of the FDA CRGC workshop on considerations for alternatives to the comparative clinical endpoint and pharmacodynamic bioequivalence studies, or BE studies, for generic orally inhaled drug products. All right, so we will begin the workshop with Symposium 1. Successes and challenges associated with the use of comparative clinical endpoint and pharmacodynamic BE studies for locally acting OIDPs. Symposium 1 will begin with introductions, followed by presentations by FDA and industry speakers listed here to discuss challenges and considerations for conducting comparative clinical endpoint and pharmacodynamic bioequivalence studies for OIDPs. This will be followed up by a question and answer panel discussion with the speakers. After that, a lunch break will follow Symposium 1. After lunch, we will begin Symposium 2, integration of alternative in vitro, in vivo, and in silico studies to establish BE for locally acting OIDPs, in lieu of the comparative clinical endpoint and pharmacodynamic bioequivalence studies. That will begin today and continue into day 2 or tomorrow. This symposium will be comprised of 3 sessions that will facilitate in-depth discussions on the various alternative in vitro, in vivo, and in silico studies that may be used for alternative BE approaches in lieu of the clinical endpoint, clinical BE studies for locally acting OIDPs. These sessions will cover the scientific and technical challenges of these approaches, along with potential ways that they may be used together for development of more complex OIDPs. Sessions 1 and 2 will run in parallel, followed by session 3. Symposium 2 will begin with a plenary talk by Dr. Brian Newman, and he will provide an overview of alternative approaches to the clinical studies for OIDPs. This will be followed up by sessions 1 and 2 running in parallel, as I mentioned before, session 1 being in here and session 2 in the other room, as Visha mentioned earlier. And we will begin today and continue into day 2. Session 1 will cover the in vitro studies that may contribute to alternative BE approaches for locally acting OIDPs, of which today we will focus on dissolution and realistic aerodynamic particle size distribution testing. And that will be the focus of day 1. Introductions and presentations will be given by those listed here. Now, session 2 will cover in vivo and in silico studies that may contribute to alternative BE approaches for locally acting OIDPs, of which alternative PK studies and in silico model selection and purpose will be the focus of today, day 1. Again, introductions and presentations will be given by those listed here. The closing remarks for day one will be provided by Dr. Markham Luke, which will end the virtual portion of the workshop for those virtual participants. For those who are actually here today in person, we will have small group discussions for those topics noted for session 1 and 2 for day one that will occur after the closing remarks. These small group discussions will be the end of day one activities for those who are here today. The outcomes of the small group discussions will be summarized and shared on day 2 of the workshop for all attendees. Without any further ado, I would like to introduce Dr. Paramjeet Kaur to begin Symposium 1. Dr. Paramjeet Kaur is a pharmacologist and senior reviewer at the Division of Bioequivalence 2, Office of Bioequivalence, in our Office of Generic Drugs at FDA. Since joining the Division of Bioequivalence in 2008, she has been involved in the assessment of bioequivalence reviews, controlled correspondences, and development of several product-specific guidances and general guidances for industry. She received her Bachelor in Pharmacy from the Banaras Hindu University in India and her doctorate in Industrial Pharmacy from St. John's University in New York. Without any further ado, welcome Dr. Kaur to begin Symposium 1.
Thank you, Elizabeth, for the kind introduction. Good morning, good afternoon, and good evening to all our participants in different time zones and from different countries. It's my pleasure to welcome everyone to Symposium 1 of the workshop. In this session, we will have 4 presentations and panel discussion. For all speaker introductions, I will be using abbreviated biographical information. For detailed bios, please refer to CRCG website. In order of presentations, our first speaker is Dr. Kiran, who currently serves as Deputy Director in the Division of Bioequivalence 3 in Office of Generic Drugs, OGD. Dr. Ren has extensive experience in assessment of generic drug submissions, covering a wide variety of dosage forms and various therapeutic areas. She has also participated in drafting of numerous agency guidances pertaining to bioequivalence recommendations. Dr. Ren holds a PhD in pharmaceutical sciences from University of Florida. Our second speaker is Dr. Stephanie Soukup, who is a physician in the Division of Clinical Review in the Office of Safety and Clinical Evaluation in OGD. In this role, Dr. Soukup is responsible for assessment of clinical endpoint bioequivalence studies, clinical consults and controlled correspondences. She also volunteers as a board-certified pediatrician in a local school-based wellness program. She received her medical degree from University of Maryland and completed her residency at Sinai Hospital of Baltimore. Our third speaker is Dr. Yu-Ching Gong, who is a pharmacologist in the Division of Quantitative Methods and Modeling, Office of Research and Standards in OGD. In this role, she utilizes quantitative tools such as population pharmacokinetics, modeling and simulations to address specific questions related to generic drug development process and/or regulatory decision-making. She is trained in pharmaceutical sciences with focuses on drug delivery, pharmacokinetics, and drug-drug interactions. Dr. Gong received her PhD in pharmaceutical sciences from University of Tennessee. Our fourth speaker is Dr. Jonathan Ward, who is Vice President and Head of Global Clinical Respiratory Program at Vitrus. He has devised and led clinical programs across all phases of development of inhaled medicines for asthma and chronic obstructive pulmonary disease, including both innovator and generic drug products. In 2011, Dr. Ward established Mylan Global Respiratory Group that became Vitrus in November 2020. He led clinical development of Vixela Inhub, a generic version of Advair Discus. Earlier, he was the clinical disease expert translation medicine leader for respiratory diseases at Pfizer and principal clinical scientist for GlaxoSmithKline. Dr. Ward holds a PhD in respiratory pharmacology from Royal Brompton National Heart and Lung Institute, London. The moderator for panel discussion is Dr. Michael Spagnola, who is a lead physician in the Division of Clinical Safety and Surveillance in the Office of Safety and Clinical Evaluation in OGD. Dr. Spagnola has led multidisciplinary review teams in the evaluation of generic orally inhaled drug products. His interests include the clinical evaluation of the user interface of complex drug-device generic combination products and retention of reserve samples for bioequivalence studies. Dr. Spagnola is board certified in internal medicine and hospital medicine and received his medical degree from George Washington University. Now I would like to hand over to our first speaker. Please join me in welcoming Dr. Kiran. Thank you, and let's enjoy the presentations.
Okay. Okay, good morning, everyone. Thank you for giving me this opportunity to present the topics on the challenges and the general consideration of conducting pharmacodynamic equivalent study for a butyl sulfate middle dose inhaler. Okay, here is my disclaimer. So this presentation reflects the view of the author and should not be construed to represent FDA's views or policies. So the presentation is structured into 4 parts. Here is the outline of what I'm going to cover. So the first, I will provide overview of FDA's recommendation on bioequivalence assessment for orally inhaled drug products. Then I will discuss pharmacodynamic equivalence for a butyl sulfate MDI as our example, along with the dose scale analysis to support bioequivalence evaluation. Then I will address the concerns and the general consideration and challenges for conducting the bronchoprovocation study with different aspects. This includes study design, how to handle the missing data in the data analysis, and the observed issue for calculate the relative bioavailability. At the end, I will conclude a summary with the key points from the presentation. So as we know, most OIDPs are locally acting drugs, which are directly deliver the drug to the site, and they will have their therapeutic effect there. Their targeted effect does not rely on the systemic exposure. So when the applicants develop the generic OIDPs, they face a lot of challenges. So OIDPs are complex dosage form, which include the formulation integrate with the device. So the performance depends on the interaction between the formulation and the delivered device, and also the performance depends on interaction between the patient and the delivered device, so including the use interface and the patient, the inhalation effort. As shown here in the figure, you can see the delivered device, the design can significantly different across the different dosage form. So the delivered device, the difference could impact the interaction between the formulation and the delivered dose and the delivered device and the patients used when they will use the drug product. Okay, to address these challenges for OIDPs,
FDA recommend weight of evidence approach. So it includes equivalence in vitro performance through in vitro testing and equivalent the systemic exposure through the in vivo PK study and equivalent of the local delivery through the comparative the clinical endpoint study and the PDB study or alternative approach, which will be covered in this 2 days workshop. And the device similates at the formulation Co1, Co2 to the reference product. So with this approach, each of these 4 components must meet bioequivalence criteria. It's important to note that the equivalent in vitro performance is not a direct indicator for the clinical efficacy. And the local delivery of the API to the site of action is a complex with multiple steps involved. And one step could in effect impact the next step. So the comparative the clinical endpoint study or PD study incorporate all the steps from actuation to the drug deposition when we evaluate whether the test product and the reference drug product have the equivalent the local delivery. So today we will focus on the PDB study. Okay, so as you can see here, there are 3 reference list drug products for the albuterol sulfate MDI. And there are several approved generic versions of the albuterol are available on the market. It's not going to. Okay. The product-specific guidance for the albuterol MDI used the weight of evidence approach was posted in 2013. In this guidance, the agency recommends 5 in vitro tests, single actuation content, aerodynamic particle size distribution, spare pattern, prime geometry, and the priming and the repriming, and the in vivo PK study, and the bronchoprovocation study. So the PD endpoint for this study is the post-dose PC20 or PD20. The analysis of the PD data is recommended use dose scale analysis, incorporate the maximum model. In order for establish bioequivalence, 90% confidence interval for the relative bioavailability should fall within 67 to 150. Okay, let's discuss bronchoprovocation study. So methylcloning is a bronchoprovocation agent that induces the bronchospasm. Albuterol, on the other hand, is a bronchodilate that provides against the methylcloning-induced bronchospasms by relaxing the muscle in the airway. The study involves administration different level of the albuterol on the different days in the crossover study, followed by a methylcloning challenge test to generate a bronchoprotection dose response curve. So the provocative concentration or dose of the methylcloning requires to reduce the FEV1 by 20% following administration of the different dose of the albuterol or placebo is used for bioequivalence evaluation. The 20% of the reduction of the FEV1 is determined relative to the ceiling FEV1 measured before administration. So since methylcloning could cause the airway narrowing, it may pose risk for the patient with severe asthma. It's important to include the subject with mild asthma as reflected in the inclusion criteria. sure how to go back to the previous slide. Okay. So the B study in PD study, the by equivalence used based on the PD study, we typically observe long-linear dose-response relationship, where the response does not increase proportional with the dose. In addition, the placebo effect could be substantial. Given this long-linear dose-response relationship, so the difference you can see in the PD, the response, may not directly reflect the relative bioavailability between the test and the reference product. To address this concern, the dose scale analysis is introduced to assess relative bioavailability in the dose scales, that will be XX, rather than the Oranger PD response scale, that's YX. So it will transfer the long-linear PD scale to the linear dose scale, allowing us to evaluate the drug at the site of action. So when you look at this curve, the dose scale analysis includes several components. First, the response from the placebo as a baseline, and the PD response from the lower and higher dose level of the reference product, and the PD response from the lower dose of the test product. For this case, it will be the 90 microgram. So using this data, we can calculate a dose response curve for the reference product. Using this curve, we can estimate the reference product's dose that could result in the response equal to the test product here. So for this case, you can see the estimated dose for the reference product is 100 microgram. So the relative bioavailability is calculated based on estimated dose divided by the actual dose. So will be 100 microgram divided by 90 microgram equal 1 to 1.11. So the relative bioavailability can be considered as the ratio of the dose of test and the reference that produce the equivalence, the PD response. Now moving to next slide. So the F, the estimate is using the E-maxima model. The detailed explanation for this equation, you can find it in the PSG for the link below. You can see for the Eurostat. So here I want to emphasize the several key points. This model assumes that both E0 and the E-maxima for the test and the reference product are the same. ED50 for the reference product is ED50. ED50 for the test product is ED50 divided by the F. The PD study design, if only includes one dose level of the test product, is acceptable. However, multiple doses of the test and the reference product could enrich the data set and increase the precision of estimated values. So the PD study is recommended use randomized at least 2 dose level of the reference product and at least 1 dose level of the test product. For the BE determination, 90% of confidence interval of the F is estimated either by bootstrapping method or we use the nonlinear mixed effect model without using the bootstrapping. Directly get the estimated F value and the standard error from the nonlinear effects model. So here is the summary of stepwise procedure for the dose scale analysis. So when we receive a data set like a 4-way, 5-way crossover, so first we can estimate the E0, E-maxima, and the ED50 from the data from the placebo and the reference product. Then we can calculate the relative bioavailability. Two methods are recommended for do the calculation of the 19% of confidence interval. So method 1 is directly obtained the 19% of confidence interval through the nonlinear mixed effect model. Alternatively, you can calculate the confidence interval through the bootstrapping. It includes 3 steps. The first step, you need to generate the sample dose response data set. The second, you need to estimate the F for each. You generate the sample dose response data set. And the next step, the third step, will be computing the confidence interval through the BCA method. You can find the detailed information in the statistical BE guidance, or was posted and revised in December last year. So for the general consideration and challenges for bronchoprovocation study, I will talk about 4 areas. One is the test batch requirement and the placebo used in the PD study, and the requirement of submission by IND, and the purpose of conducting pilot study, and how to handle the missing data during the study, and the issues we observed during the calculation. So for you, in general, for the in vivo study, the batch requirement for the tester and the reference product should be the same for the in vivo PK study and the PDB study. Those of the batch should be the one of the 3 batches used for the pivotal in vitro BE studies. So if the test product lot will expire, and additional test batch has to be manufactured to complete the PDB study, the following information is recommended. So the new test batch should be manufactured, use the same equipment under the same manufacturing process. The applicant is recommended to submit CMC information for this batch. Regarding the batch size for this additional test batch, please follow this guidance list here for the batch size. Regarding the placebo information, during our IND reviews, some applicants did not specify the composition of the placebo. So the formulation of the placebo should be the same as test product, except without API. So the by-IND is required to submit to the agency before conducting the PDB study. As the methylclonin concentration exceeds the FDA-approved amount, 25 milligram per milliliter, particularly at the high dose of the butanol. Here is the 0.18 microgram, where the 25 milligram per milliliter of the methylclonin may not achieve 20% of the reduction of the FEV1. So applicants are encouraged to conduct a pilot study to refine their study design, including the inclusion and exclusion criteria, estimate the study power according to the intra subject variability, and we can estimate the slope of the E-maxima model. So moving on to another challenging issue is the missing PC20 data in the bronchoprovocation study. When it comes to the missing PC20 data, there's no consensus how much % of the missing data is considered acceptable and will not impact on the bioavailability the evaluation. How should we, let's look at the scenario number 1. So PC20 is the methylclonin concentration required to reduce FEV1 by 20%. Sometimes subject receive the maximal concentration of the methylcloning, still cannot achieve 20% of reduction of FEV1. How should we handle this situation? For example, some applicants choose to impute the unmeasurable value to the maximal concentration of the methylcloning at the test and the referenced treatment period. By using this approach, it could decrease the PC20 difference between the test and the reference product. Substantially, it could bias the BE evaluation. Our recommendation is that if you're going to choose imputation approach, please specify your imputation strategy in your statistical report, supported by the adequate justification. It's recommended to you to evaluate whether the proposed imputation approach is validated and is sensitive to detect the product difference. I'm moving to the next page. Okay, so missing data scenario 2. So data in some subject treatment arm may be missing due to protocol deviation, such as the subject did not complete this period due to the side effect, or the subject withdrawn the consent forms, or some dose error. So in the table you can hear, this is a 5-way crossover study. The red front represented the missing period. Since we know the missing PC20 data could potentially bias after assimilation, the applicant is recommended to include their justification to evaluate whether this missing data could bias after calculation. Okay, in terms to the bootstrapping, during our review evaluation process, we have seen the multiple or different way to implement the bootstrapping. Because this is a crossover study, one subject has multiple of the observation from the different treatment. So here I'm going to show some example of different way of the bootstrapping. Okay, so the table 1 shows the orange data from the PD study for the 3 subjects in the 4-way crossover study. The table 2 shows the sample resampled by the observation without preserving the subject information. So the table 3 is the sample by resample by the subject, but only with the reference product. Oh, it's cannot come back. So the last table, let me go back. So the last table is the sample by resample by the subject. Among those 3 method of the bootstrapping, we recommend the last one because the bootstrapping sample unit should be subject. Use this way, you can maintain the correlation of the observation with the subject. In summary, bronchoprovocation study can be used to demonstrate the PD bioequivalence for the albuterol sulfate MDI with a non-linear dose-response relationship. For reliable the dose scale analysis, the applicants is recommended to have appropriate the study design, which including suggest conducting a pilot study and stating how missing data will be evaluated, incorporated in the study protocol, and the sufficient justification for evaluating the missing strategy on the PC20 data should be provided in the submission. As we know, given the complexity of the dose scale analysis for PD study, early communication with agency is encouraged to discuss your proposed study design and the statistical alternative statistics approach. So acknowledgement. I would thank you, Pastor April, Bing, Tian, and Diana, Brian, Lamfeng, and Liang, and Men to help me to develop these slides. And thank you for your attention. Sorry for the technical problem.
Morning, everyone. Okay, excuse me, let's get started. Before we move on, let's see. It's not advancing. There we go. Just a quick disclaimer before we begin. There we go. Okay. So in today's presentation, we'll define a comparative clinical endpoint bioequivalence study, introduce challenges and limitations of these studies for dry powder inhalers, discuss how a detailed study design can avoid common confounding factors and result in higher quality data. We'll utilize an example to illustrate how challenges can be mitigated with a good study protocol. And we'll talk about some recommendations to consider in your study protocol. Okay. So comparative clinical endpoints bioequivalence studies are appropriately designed comparative clinical trials conducted for the purposes of demonstrating bioequivalence. More specifically, they're clinical studies in a patient population where two pharmaceutically equivalent products are administered, delivering the active moiety to the local site of action. These studies are used in 1, dosage forms with a local site of action, 2, those not intended to be absorbed, and 3, in drug products where the traditional pharmacokinetic studies are not feasible or sufficient alone. So in DPIs, a clinical endpoint bioequivalence study is recommended because of uncertainties in the correlation of in vitro and in vivo PK data to the clinical effects at the site of action.
Yeah, you can say just next slide and we can change it from there.
Next slide, please. Next. Okay. All right, so there are several challenges in these studies for DPIs. First, they're pretty expensive. Next. This is due to the larger population required and also requiring multiple clinical sites. And also because they tend to be more lengthy than other BE studies, such as PK studies. Next. Unfortunately, these challenges are a bit harder to avoid. Next. There are also several challenges that are more unique to studies in DPIs. First is spirometry. Differences in spirometry equipment and variability in subject technique can influence your results. These studies can also be greatly affected by subject behavior and reporting. For example, accurate and complete records of rescue medications, subject retention and dropout, as well as how well subjects document adverse event symptoms and study medications. These are all areas for deficiencies. So the evaluation of comparative clinical endpoint bioequivalence study data and study results are largely affected by how the study was designed and conducted. A good study design can address many of the challenges and confounding variables inherent to these studies, avoid deficiencies in multiple rounds of information requests, and ultimately result in higher quality, less biased data. So, when designing your study, start with the product-specific guidance and follow the recommended study design. Also, provide extensive detail in the study protocol on how your study will minimize limitations unique to the study. For example, how spirometry data will be collected, but also how subject behavior can be accounted for through specific instruction and counseling. To illustrate some of these challenges and issues, let's use an example. The RLD will be Advair discus. As we know, one of the indications for this product is the treatment of asthma in patients aged 4 years and older. It's given as 1 inhalation twice daily. Next. In this case, there is a PSG. It recommends several studies, one of which is the comparative clinical endpoint bioequivalence study. The PSG details a 2-week run-in period followed by a 4-week treatment period comparing the test, reference, and placebo in subjects with asthma. There are several additional details for the study, including those for spirometry collection, as well as a list of prohibited medications that may mimic the effects of either the LABA portion of the drug or the inhaled corticosteroid. The co-primary endpoints are area under the serial FEV1 time curve from time 0 to 12 hours, and also to compare the change in FEV1 from baseline to day 28. And each endpoint compares one active ingredient of the test product to the RLD. So let's take a closer look at a few of these challenges. The first is spirometry. For studies in DPIs, spirometry is used to evaluate subjects for eligibility into the study as well as the primary endpoints. There's potential for inaccurate or inconsistent measurements either across sites or for each patient over multiple visits. So the protocol has to include detailed information on all equipment and procedures involving spirometry. So in designing your protocol, carefully detail definitions and procedures for how FEV1 measurements will be taken, such as what equipment and software will be used, and whether that'll be the same across sites. Document what calculation for predicted FEV1 to determine eligibility into the study will be used. And just a reminder, this calculation should be the same across all sites. Also, define what's considered an acceptable reading. Will this be determined by the software, a third party, etc.? And specify how many recordings will be taken for each spirometry measurement. For example, at baseline, will subjects be given an opportunity for a minimum of 3 attempts and a maximum of 5, that level of detail. So going back to our example for a fluticasone and salmeterol product, the protocol may state that all clinical sites will be provided by the same with the same spirometry equipment and software. The protocol would also provide the exact formulas and provide an example calculation. Our next challenge is rescue medications. Because comparative clinical endpoint bioequivalence studies for DPIs are conducted in patients, rescue medications are necessary. Typically, they're short-acting beta agonists. Rescue is permitted throughout the study for subject safety. However, the use can potentially bias study results, so clear limits have to be pre-specified in the protocol. For example, rescue within 6 hours of spirometry measurements. So in designing a protocol and instructing subjects on the use of rescue, be very specific on how subjects are going to be instructed to use their medication. This should always be for symptoms rather than on a set schedule. Also document, go back. Also document when to contact study personnel. This could be if subjects have used a certain amount of rescue within a pre-specified period of time. Document whether rescue medication will be provided or what medications are acceptable for use as rescue. And lastly, subject recording of rescue medications, including the date and time of use and a clear instruction to avoid rescue within 6 hours of any FEV1 measurements. For rescue data sets, they can be submitted as their own rescue data set or within the concomitant medication data set. But if this option is chosen, each rescue use has to be clearly marked as rescue so it can be differentiated from other concomitant medications. Bringing things back to our example, the protocol could include the following statements. The first statement notes which medication will be used as rescue and provided by the study. The second clearly restricts the use of rescue within 6 hours of FEV1 measurements. For fluticasone and salmeterol product, remember that FEV1 is measured at screening for inclusion into the study, at baseline, serial measurements on day 1 to assess salmeterol, and then again at day 28 to assess fluticasone. Taking rescue before screening can result in inappropriate inclusion into the study. And rescue before baseline, day 1 or day 28 measurements could alter one or both endpoints for that subject. The third statement is an example of what the subject is expected to do. In this case, it's a detailed recording of all rescue medication use. This is helpful to ensure that subjects have not used medication within that 6-hour window before study visits, and also may help determine treatment failures or inappropriate rescue use. An additional prompt for study personnel to specifically ask and document rescue medication use before FEV1 measurements is also helpful to verify that they've asked this information and document. And this documentation should be in the case report forms. Moving on to concomitant medications. Any meds with anti-inflammatory effects, such as steroids, can affect FEV1 measurements and introduce bias into the study. Others can as well. So all medications have to be recorded, whether or not they're related to the study drug or the condition that the study drug is intended to treat. and this includes as-needed medications. As a reminder, reference the PSG and start there. So in your protocol, include a detailed list of all medications restricted during the study. I can't stress enough to start with the PSG. Also, consider any other medications that may affect eligibility or the evaluation of one of the primary endpoints. Also, specify how medications will be recorded and include the date and time for any meds. In our fluticasone and salmeterol example, the PSG referencing Advair includes a list of restricted concomitant medications. The protocol for the proposed product should include all of those as a minimum. The protocol would also include how concomitant medications would be documented. The first statement reflects how the subject would record those concomitant medications, which is the subject diary. If study subjects document any medications in their diary, the investigator would then check the diary, ask about any additional medications, and record all of the answers in the case report form. So moving on to our next challenge is treatment failure. In any clinical study, we expect to have some subjects with lack of efficacy or treatment failure. Failing to identify treatment failures can bias the results, but also result in the loss of evaluable subject data. Subjects with adverse events or symptoms of underlying disease exacerbation can represent treatment failures, so consider these. Treatment failures should also be included in the statistical analysis. Therefore, a clear definition should be pre-specified in your protocol. So a few recommendations. Specify how and when to instruct subjects to contact study personnel. This could potentially be tied to rescue medication use, but also if an adverse event occurs that's consistent with symptoms of an exacerbation of the underlying disease, this could also represent a treatment failure. If subjects appropriately contact the study personnel, they can be evaluated for those symptoms, and if appropriate, the investigator can then initiate end of study procedures and include that subject in the statistical analysis as a treatment failure. Consider subjects' reasons for discontinuation as well. Any information about adverse events, requirement of additional medication, or poor symptom control are extremely important to capture and should be included in the case report forms and whatever applicable data sets. Lastly, consider the treatment day and number of the doses that the subject has used, because many DPIs aren't expected to reach their full effect within the first several days of treatment. For our example, a possible definition of a treatment failure noted on the screen addresses reasons for discontinuation, requirement of additional medications, but also includes language regarding the number of received doses specific to a fluticasone and salmeterol product. It also states that subjects will complete the specified end of study procedures and included in the per protocol population. Okay, and just a few last recommendations for your study. The first is to carefully define your per protocol populations and ensure that they're consistent with the PSG if one is available. Second, document the narratives for any adverse events, especially SAEs, as well as the reasons for subject discontinuation, both related or unrelated to exacerbations of the underlying disease, such as asthma. These should be documented in the case report form. This step can often reduce the number of IRs for clinical concerns. And lastly, think about treatment compliance, subject medications, and adverse events when designing your subject diary, because this can help capture that desired information. There we go. So on the screen is an example of a subject diary entry. It includes the study day, also has spaces for the study treatment dose to check off if taken for compliance, spaces to document concomitant meds, both daily or as needed, as well extra spaces just to ensure if somebody has to start a new medication that they didn't anticipate, we want to capture that information. And also include spaces for rescue medication doses, times, and the symptoms. Also, consider how to record additional medications to capture all doses in case there's a long list of those additional medications. But also consider a prompt of how and when to contact study personnel that's consistent with your study's definition of an adverse event, exacerbation, or a potential treatment failure. So to wrap up, comparative clinical endpoint bioequivalence studies can be limited by expense, time, and several confounding variables. However, careful study planning and detailed documentation can help mitigate some of these challenges and ideally end in performing the study only once. Also, for specific questions and feedback, I encourage you to consider the controlled correspondence and pre-ANDA meeting pathways. Thank you.